I finished talking about ribonucleotide reductase and the regulation of that last time, and I also talked about the metabolism of thymidine. So thymidine metabolism, of course, derives from uridine metabolism, and I showed you all of that. One of the things I didn't get to show you was this right here, and that is the reaction in which DUMP is made into DTMP. I showed you that there was the addition of a methyl group. Hear a lot of noise, folks, please. Shh. Where there was addition of a methyl group, I didn't tell you where the methyl group came from. So the methyl group comes from a molecule known as folic acid or folate. Okay? And so folates, and there are a variety of folates, are very important in what we call one-carbon metabolism. And we can actually see that carbon right there in that little oval that is not present in this structure over here. And what's happened is that carbon became that methyl group right there. So folates are important sources of single carbons in metabolism, which is why we call them one-carbon metabolism molecules. The reaction here is catalyzed by the enzyme known as thymidylate synthase or thymidylate synthetase. I use those two terms interchangeably. And there is a molecule that is a suicide inhibitor of that enzyme. It's known as 5-fluorouracil, and it is an anti-cancer treatment. And this makes really good sense because what do cancer cells have to do? They have to divide. And if they have to divide, what do they need? They need nucleotides. Specifically, they need deoxyribonucleotides. And if they can't make thymidine, they're hosed. So inhibiting this enzyme is one approach to stopping cancer cells. The downside, of course, is that when you make any inhibitor of deoxyribonucleotides, you affect normal cells as well. So the strategy of any of these is that if the cancer cell is dividing more rapidly, it's much more sensitive to and much more needing of deoxyribonucleotides than a normal cell is. And so consequently, the idea is that you kill the cancer cell preferentially. And one of the side effects that you frequently see with this approach, of course, that people get very ill, lose their hair, and so forth. And that's because you're killing a lot of normal cells in the process. And it's not unique to this drug. It's a common problem associated with trying to specifically kill cancer cells. Now, looking at this in another level, there's a very important consideration. And the important consideration is that just like the cell has a limited supply of NAD/NADH, so too does the cell have a limited supply of folates. So if I take that methyl or that carbon off of that one folate, I have to be able to regenerate that folate with that carbon, because if I don't, all of the folates will end up minus that carbon. And if everything is minus that carbon, I can't make thymidylate nucleotides, okay? So that recycling is very important, and the recycling, at least part of that process of recycling, is shown right here. We went from this mouthful of a molecule name, and no, I'm not going to ask you to know that molecule name, and produced a molecule called dihydrofolate. Dihydrofolate is a molecule I think you should know, okay? It's the product. What do you get after you pull that carbon off of this monster folate molecule? You get dihydrofolate. And the reason I want you to know that name is because the enzyme that is involved in this recycling process that's relevant for our purposes is called dihydrofolate reductase. You can call it DHFR. DHFR converts dihydrofolate into tetrahydrofolate. And I don't even care if you know that. I just want you to know that this enzyme is important in this recycling process. You can actually see the gain of the extra carbon in going from tetrahydrofolate up here, but that's not my reason for showing you this figure. My reason for showing you this figure is that this enzyme, dihydrofolate reductase, is the target of another anti-cancer drug, and that's called methotrexate. Okay? So methotrexate, I have, I think, briefly mentioned before, and I'm sure Dr. McFadden probably mentioned it last term, is a competitive inhibitor of DHFR, meaning that it looks an awful lot like the dihydrofolate substrate. It looks like it enough that it binds to the active site of the DHFR, but the DHFR is unable to handle it, and consequently the enzyme is inhibited. Okay? So that means then that methotrexate is another approach to an anti-cancer drug. It's not a suicide inhibitor, it's just simply a competitive inhibitor. Remember the difference between a suicide inhibitor, which covalently binds to a molecule, and a competitive inhibitor, which simply binds but not covalently, okay, is that a competitive inhibitor can come off, right? So we're not killing all of the enzyme by adding methotrexate. Well, that turns out to be a little bit of a strategy for the anti-cancer use of methotrexate. Because if you wanted to really kill selectively cancer cells that were dividing very rapidly, you could dose a patient with a fair amount of methotrexate and then withdraw it. And when you withdraw it, hopefully you're going to rescue the DHFR in the healthy cells after you've already killed the cancer cells. So that's one strategy, or it's another strategy for using chemotherapy to kill cancer cells. So methotrexate's a very important player in that process, and the important part is it's inhibiting the recycling of the folates in this overall process. Okay. One last note there, and again, this is an enzyme, you don't need to know the name of it, but I just point out for anybody interested in health considerations, and that is that the Plasmodium enzyme, which is the organism that's involved in malaria, has a different enzyme from the human and can be targeted with anti-malarial drugs. Yes? How would you withdraw the molecule? Yeah, to be honest, I'm not a physician, so I probably couldn't tell you, but imagine that you injected, for example, the methotrexate at the site of a tumor, and then over time, that washed out. That would be one approach. I don't know actually how it's done myself, but that'd be one way to do that, because over time, the concentration of it is going to dissipate. Okay? Okay. Other questions? So, knowledge of the nucleotide metabolism helps us to better understand approaches to treating cancer. Okay, and there is the comparison between the structure of methotrexate and the structure of dihydrofolate, and you can see that they are very, very similar. It's no surprise that they might both bind the active site of the DHFR. Okay, the last thing I want to talk about are the use of nucleotide analogs. Okay? We've been moving in the direction of therapies and ways of treating disease and so forth, and so there are a ton of nucleotide analogs that have been developed in recent years to treat diseases like Ebola, like HIV, like other viruses, or in some cases, rapidly dividing cells like we would have in a cancer. There are basically two approaches that I'll talk about that have been used in the design of these drugs. Okay? But before I talk about the design, I've got to talk about a consideration of getting drugs into cells. Because if you think about it, we have to get these compounds across a lipid bilayer if we want them to, for example, in the case of a cancer cell, we want to be able to get it into the cell to do something. And so that's not a trivial thing. You've seen that in many cases, movement of molecules across membranes requires membrane proteins that are specific for the molecules being transferred. Well, it turns out that's a consideration here as well, because if we compare AMP, which is this guy here that has a phosphate, with adenosine, the nucleoside that lacks the phosphate, there's a very big difference in the ability to get these two across a cell membrane. Cells will transport this guy on the right. They will transport the nucleoside. They will not transport the phosphate part of that. So all of the compounds I'm going to show you are made in the nucleoside format. But that means that once they get into the cell is going to have to use the salvage pathway to make the corresponding nucleotide if it's going to be effective. Okay? So the use of these compounds in a cell rely on two things. One, getting across the membrane, and two, relying on the salvage pathway to phosphorylate it so that it can be used. Well, obviously, if you phosphorylate adenosine, you make ATP, so there's no drug, right? That's the normal substrate for RNA polymerase to make RNA, right? However, there, as I said, are two approaches to the design of these drugs that allow cells, or allow physicians treating patients to stop either replication of DNA or replication or the synthesis of RNA. And the two approaches I'm going to tell you, and then I'm going to show you some examples, are as follows. One is to design a competitive inhibitor of either the DNA polymerase or the RNA polymerase, meaning we make a nucleotide-like molecule that fools the polymerase. Okay? If the target is, let's say, a plasmodium organism, that we get it into the cell, we want to be able to target, okay, that replication system in that organism. So we want to make something that looks really a lot like the normal nucleotide, but that is fooling the enzyme. So a competitive inhibitor is one choice for that. A second choice is something that the polymerase will actually use, but when it uses it, it makes something that can't be further made into a nucleic acid. Okay? These are what we call chain terminators. Let's think about what, we're going to talk later today about DNA polymerase, but DNA polymerase makes a DNA strand by copying the other strand and putting nucleotides together and making phosphodiester bonds to join them all together. What if instead of continuing that process, I put nucleotides in, I put nucleotides in, I put nucleotide in, but then I put in a nucleotide that something can't be attached to? Right? What that means then is that this is what we call a chain terminator. Something can't be attached to it, so I can't make it any longer, and if I can't make it any longer, I have stopped either the DNA replication or the RNA synthesis for that target organism. That turns out to be really, really useful. Okay? So the two approaches, competitive inhibition and chain termination. Let's look at these guys. First of all, we notice when we look at these derivatives that are made, here's adenosine, a normal thing that a cell might have inside of it. Here's a couple of molecules. I'm not going to ask you to know the names of these, so don't worry about the names, don't worry about the structures. But the structures have some interesting features. Look at this guy here. It has no 3' hydroxyl. The guy on the left has no hydroxyl. Well, it turns out, as we'll see today, that a 3' hydroxyl is necessary for something to attach to continue growing the chain. The phosphate, this gets phosphorylated, the phosphate can get attached to the previous nucleotide, but the next nucleotide has nothing to attach to. Therefore, this guy on the left is a chain terminator. Once it gets built into the nucleic acid by the polymerase, in other words, the polymerase recognizes it just fine. Once it gets put in by the polymerase can't add anything to it, and you've stopped the synthesis of that nucleic acid. Okay? On the other hand, this guy on the right has a 3' hydroxyl. If the polymerase puts this in, okay, it does just fine, but this guy is a competitive inhibitor. This guy binds preferentially to the right nucleotide and inhibits the enzyme. So this guy will actually stop the enzyme from functioning before it ever gets to the point of putting it into the nucleic acid. So it's inhibiting the synthesis before this even gets put in. This guy gets put in and stops. This guy inhibits the synthesis and getting put in. So the guy on the right inhibits the polymerase. The guy on the left inhibits the polymerase from acting further. And so that's the strategy of all these. And I'm just going to briefly flick through them and show you some of them. Some of the designs are very similar to nucleotides. Some of them are different enough that you'll be surprised that they actually do work. Okay? This guy here is a very subtle difference in the base. Look at that. Okay? Very, very subtle difference in the base. It's used to treat Ebola. And again, you don't need to know the names or the structures here. Look at these guys, okay? So you could look at these and say, well, that right there is probably a competitive inhibitor because it's got a 3' hydroxyl. Everything else looks like it's okay, okay? You look at this guy over here and you say, whoa, that's probably a chain terminator because there's no hydroxyl, there's no place to put something on, right? There's a chain terminator. And God, I don't even know what that one is. That one is so weird, all right? I would imagine it's a competitive inhibitor, but I don't even know, okay? But it works, okay? It works. And it works because through some mechanism, either through chain termination or through competitive inhibition, it's stopping the synthesis of the HIV virus. The advantage of the HIV virus inhibition is that HIV virus has its own DNA polymerase that has slightly different specificities than the normal human DNA polymerase. So the idea is that you can specifically target HIV DNase polymerase, it's called reverse transcriptase, and hopefully have very little effect on the normal cellular polymerase. And that's the strategy that's used in a lot of the anti-HIV drugs that are there. Okay, questions on that before we move on, finish? Okay, then let's move on to DNA replication, recombination, and repair. The 3 R's, replication, recombination, and repair. This is what we've already seen, right? The difference between a ribonucleotide and a deoxyribonucleotide simply relates to the fact that a ribonucleotide has a hydroxyl at position 2 on the ribose. Deoxyribose contains a hydrogen there instead of a hydroxyl. That very minor difference has very big implications. One of the big implications, DNA is a lot chemically stabler than RNA. The presence of that hydroxyl right there contributes to the chemical instability of RNA compared to DNA. So when you hear about researchers that have found some sample that's been sitting around for a few hundred or a few thousand years and they are able to test it, they're testing for the DNA because the RNA that's in there has long since been demolished. Okay? So DNA is chemically stabler than RNA. Okay. You've seen DNTPs, and you know DNA is made up of the starting materials are DATP, DGTP, DCTP, and DTTP, and I use the D, of course, to indicate the deoxy form of each of those. Yeah, somebody, oh, I feel that sentiment. Oh. Okay. DNA has specific structure that we refer to as having polarity, meaning that we can tell one end of it or one side of it from another. DNA, of course, you know, is a double helix, most commonly in cells. But if we take the strands apart, we end up with a single-stranded DNA, and that's what's happened here that you can see. I like this figure because it schematically shows you the chemistry of a DNA strand. Let's talk about those individual points. First of all, there's what I call a polarity to it, meaning that there's an end. You can distinguish one end from the other. At one end, we have what's called the 5' end. That's this phosphate hanging off right here. It's named for the fact that that's carbon number 5 of the ribose ring, or the deoxyribose ring, and nothing is attached to it except a phosphate. This guy has an attached phosphate, okay, and is already linked. So the end, in this case, is the free end of it. At the other end of the molecule, we see a free 3' hydroxyl. And you notice that in making this, we are joining the 3' end of one to the 5' of the next, the 3' to the 5', the 3' to the 5'. We say that the direction of synthesis is not 3' to 5'. We actually say it's from the end moving forwards, meaning it's 5' to 3', even though we're joining 3's and 5's. So the direction of synthesis, and this is one of those absolutes. You see very few absolutes in biology, but this is an absolute. You only see the synthesis of nucleic acids in the 5' to 3' direction. Only. Absolutely. Okay? Don't forget that. What else do we see here? Well, we see these phosphates are the links between the individual nucleotides. These are what we call phosphodiester bonds. Okay? And these phosphodiester bonds are joining adjacent deoxyriboses. Notice the phosphates aren't even touching the bases. Okay? So we could think of this as phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, and then sticking off of each sugar is a base. Okay? We will see later that when a double helix forms, the bases end up being on the inside of the double helix, and the phosphate sugars end up being on the outside. And that makes very good sense for a cell. Because, why? Bases are relatively nonpolar. They don't like water. They like to associate with each other, whereas phosphates and sugars love water. They're very hydrophilic, and they're on the outside where they can associate with water. You saw proteins fold themselves in that way, so that the proteins folded so that the hydrophobic amino acids tended to be on the inside, and the hydrophilic amino acids tended to be on the outside. Same thing holds for nucleic acids. Okay, let's continue with this idea, okay? This says helix, although it doesn't show much of a helix. It just shows the two strands paired to each other. But if we look at the two strands paired to each other, we see that in the middle of the two strands, we have the bases arranged with each other. And of course, every grade school kid almost anymore learns that A pairs with T and G pairs with C. The forces that hold the strands together are hydrogen bonds between the bases. The forces that hold the strands together are hydrogen bonds between the bases. AT base pairs, adenine, thymine, have 2 hydrogen bonds that hold them together. GC base pairs have 3 hydrogen bonds that hold them together. That means that if you're trying to break them apart, it takes more energy to break apart GC base pairs than it does AT base pairs. And we'll see that's important when we go to examine the process of transcription, because in transcription what a cell has got to do is it's got to separate the double-stranded DNA and start making RNA. We'll see how that comes into play later on. Okay. Another thing that you see here is that the strands are oriented in a fashion that we call anti-parallel. That means that the 5' end of one strand going down to the 3' end of the same strand is exactly opposite of the 5' end of the other strand going upwards towards the 3' end of the other strand. They're arranged like this instead of being arranged like this. Make sense? Anti-parallel orientation is important. Now, there was something I mentioned earlier. I'm going to come back to this numerous times because it's so important, and students memorize it but don't learn it. Right? There's something that you memorize but you don't learn. Okay? I find this happens all too often. Memorizing something means that you can regurgitate it, but learning it means that you can use it. All right? So I'm going to tell you something I want you to learn and not memorize. And that is that all nucleic acid replication occurs in the 5' to 3' direction, and there are no exceptions. Well, I got that. No, you memorized that. I'm going to show you in just a little bit why you memorized that and how you haven't yet learned that. Okay, will you trust me on that? Hell no, you're the instructor, right? Okay, so anti-parallel, and we remember that the replication is always 5' to 3'. Well, now we see more of a real live double helix. This is something we refer to as the B form of DNA. The B form of DNA is the most common form of DNA that's found in cells. It was the form of DNA that was reported by Watson and Crick in a very famous paper in 1953. Watson and Crick were able to use the data of a woman named Rosalind Franklin that they essentially stole to discover this structure, and they got all the credit. And she got credit for a minor contribution when, in fact, she had the major thing here. She actually found in the very same issue of Nature, and she reported another structure of DNA that at the time was considered to be not important. And in terms of percentages, it's not the most important structure, but it's called the A form of DNA. I'm going to show it to you in just a second. At the time when she reported, it was thought that the A form of DNA didn't occur in cells. The reason why was, in order for her to study those, she had to make very, very dry structures. And the only way the A form would form was in these dry structures. And people said, well, cells are full of water, so this isn't important. Well, we now know that the A form does, in fact, form inside of DNA to a minor extent. And so Rosalind Franklin was the discoverer the sole discoverer of the A form of DNA. She was a contributor to the B form, but she didn't get credit, unfortunately. Well, if we look at the B form of DNA, we see those strands that are intertwined with each other. And we see something called a major groove and a minor groove. And I've schematically shown them on here. And no, I'm not going to ask you to draw that on an exam. I just simply show you this to refer to these structures, because when people talk about the structure of double helical DNA, you will usually hear major groove and minor groove. And this is what it's referring to. This portion being the minor groove, because you see a smaller gap between the strands, and this portion being the major groove, where you see a bigger thing between the strands. Proteins that bind to DNA will tend to bind to either the major groove or the minor groove. That's the place where they will typically bind. Okay? So major groove versus minor groove. And we see major groove versus minor groove in other forms of DNA that are most pronounced in the B form, which is the form that you see here. Another feature of the B form of DNA is the number of base pairs per turn of the helix, meaning when the helix repeats itself, like I started here, I go up here and repeats itself here, I will have found that there's 10.5 base pairs. Meaning if I wanted to get an even number, I'd have to go 2 full turns to get 21 base pairs of DNA. So there's 10.5 base pairs per turn of B form of DNA. Very important number that turns out that we'll see a little bit about later. I mentioned the polarity before, and this figure simply shows you the same thing in another way. That is that the 5' to 3' orientation of the top strand goes from right to left, whereas the strand it's paired to goes from left to right in the 5' to 3' direction. And no, we don't have to have an end to figure out the polarity. We can figure out 5 to 3 by simply looking at the strand. All right, I mentioned the A, B forms of DNA. There's a third form that we talk about called the Z form of DNA. The B form of DNA is the one I just showed you. The A form of DNA is the one I said that Rosalind Franklin discovered. If we look at the A form of DNA, it's a little hard to look at these structures and pull something out, but I'm going to tell you the sort of salient features of them right now. Okay? This guy's the most common. The middle one's the most common one we find in cells. Probably 99% of the structure of DNA in cells is in the B form. Okay? The A form, as you can see, is more compact. And the A form also tends to form a bit of a bend. It's not real apparent here. But where A structures appear in DNA, we see bends. And we think those bends may be important because DNA has got to fit into a cell. And that might seem, well, of course it fits into a cell. Let me give you the consideration about that that's important, all right? If you take all of the DNA in one cell of your body, all the DNA in one cell of your body, and you stretch it end to end, it's 7 feet long. That cell is smaller than your eye can see. How do you fit it into there? Well, it turns out you've got proteins called histones that help to wrap up and coil up DNA. And it may be that some of these bends that the A form of DNA have help in that process of bending and fitting things in. But that 7 feet of DNA is fitting into a nucleus that's not much bigger than a bacterium. Okay? That's mind-boggling. Another mind-boggling thing is if you take all of the DNA of all of your cells and you stretch it end to end, you have enough DNA to go to the sun and back. You have 180 million miles of DNA walking around with you every day. Do you feel tired some days? All you have to do is, I've got 180 million miles of DNA I'm responsible for, guys. I'm taking care of all this stuff, right? No wonder you're tired. 180 million miles of DNA in your body. It's pretty mind-boggling, right? Pretty mind-boggling. Okay, well, what about the Z form? The Z form of DNA, when it was first discovered, it was kind of like the A form of DNA. They said, this thing can't be real, it's an artifact, okay? Because they found it by messing with some stuff in a laboratory, okay? Kind of like what Rosalind Franklin was doing. And they found this structure. Well, this structure looks different, but it's even more different than it appears on first glance. I'm going to tell you some of the significant differences about DNA. And yes, we know zDNA also appears in cells. Okay? The first difference between the A and the B are similar. Okay? All three of these have anti-parallel strands. All three of these have AT base pairs and GC base pairs. All three of these have phosphodiester bonds, linking things together. All three have the bases in the middle. So what's different? Well, this guy has a very pronounced major versus minor groove. We don't really see much of that there. If we look at the distance between the bases and we put a ruler up there, we would see that the Z form is more stretched out. The bases are further apart. But the most important thing between these is that the A form and the B form coil the strands in the same direction. They're what we call right-handed helices. Right-handed. Now, that's difficult for me to convey to you here, and I'm going to ask you to trust me. I'm not going to ask you to recognize the difference between a left and a right-handed form. If you would like to see an example of coiling, come to my office and I'll show you how coiling, can be done in a right or left-handed fashion. A and B form DNA have right-handed helices. Z form DNA, you probably see this coming, has a left-handed helix. So that means that these strands are going this way in the right-handed form, and they're going this way in the left-handed form, and both of these structures can appear within a chromosome. Now think about that. Imagine that you've got a section of chromosome where it's winding here, and then it has to go the other direction for a ways, and then it goes back to the right-handed form. That's actually what happens inside of a chromosome. Okay? Why would the cell, or why would the DNA have both of these things? Well, it's actually built into the chemistry of the DNA. And one of the things that we see about cells, one of the things that cells do, okay, is they use everything to their advantage. And they use this to their advantage as well. How do they use that to their advantage? Well, you might imagine that if you coil, and then you uncoil, and you go back and you coil again, that might look different, right? Cells use that. Cells, we think, actually use zDNA as a flag that says there's a gene nearby. There's a gene nearby. Why is that important? We got 7 feet of DNA. The average size of a gene is smaller than your eye can see. A lot. You hear the thing about eukaryotic DNA, which is the DNA that we have. Most eukaryotic DNA is what people used to refer to as junk DNA, meaning most of the DNA is not in the form of a gene. How does the cell know where the gene is? This may be one flag that tells the cell where the gene is. It's structurally different. Now, what I'm telling you here was actually, I'm not going to go through the whole story with you, but was actually discovered by an undergraduate working here at OSU doing undergraduate research when he was a sophomore. Now, I tell that as an example to students, okay, because I want every student to get involved in undergraduate research. And they think, oh, that's for super brains, that's for XYZ, et cetera, et cetera. That sophomore was not as far along as you are, doesn't know as many things as you know, okay, has no more skills than what you do, and he was able to show that these structures are very close to genes. Pretty cool, right? Get involved, okay? Get involved in undergraduate research. If you're not doing that already, go work with a professor. You'll learn more working with a professor than you will listening to me, I can tell you that, okay? Very cool thing to do. Okay, so anyway, zDNA turns out to be pretty important, we think. It may have other functions, it may do other things, but one of those may be flagging where genes are. Okay. DNA replication, that's our topic for the next couple of days. And DNA replication, of course, means that something that starts out as 2 strands has to be made into 4 strands. And those 4 strands have to separate because that's the genetic information for a cell. In order for a cell to divide, the DNA must replicate. One of the first things that people wanted to know after they determined the structure of DNA was, how does DNA replicate? Because we know it has to replicate because it has the information necessary for a cell to make everything that it makes. So I can't have a cell without DNA, so if I have cell division, I therefore must have DNA division. Well, DNA division happens as a result of DNA replication. And DNA replication occurs through a process that we refer to, or that we describe as semi-conservative. Semi-conservative, okay? Semi-conservative means exactly what you see on the screen here, meaning that each replication of DNA results in 2 molecules, one molecule that has 1 strand from the parent and 1 strand that is newly synthesized. The other strand has 1 strand from the parent and 1 strand that is newly synthesized. And you might say, well, duh. Well, before they found the enzymes that did this, nobody knew if that was the case. We knew it replicated, but you could have replication in which case you have one new one that has both strands of the parent and the other one that has 2 new strands. You didn't know how the copying was done. You didn't know it was copied directly from the strand. We now know, of course, this is the case. Semi-conservative replication means what I just told you, and it occurs by the reading of 1 strand by a DNA polymerase to make the other strand. Okay? That's what happens during semi-conservative replication. We're going to see here this thing on the left that's interesting. We see that the synthesis of these two progeny strands occur in very different ways. We have what we call leading strand synthesis that occurs continuously, meaning we make one uninterrupted piece of DNA. And the other strand that's being made is made in what we call the lagging strand synthesis, and it's discontinuous. It's made in pieces. And you might say, why does the cell do that? And I'm going to show you why the cell does that in just a second. This schematically shows you the semi-conservative replication, and it shows you the leading strand synthesis going on this top strand labeled B, and the lagging strand synthesis occurring on the bottom strands labeled C. And you'll notice that the 2 strands on the bottom strand are in pieces. Now, why in the world does the cell complicate matters for itself? Well, let me show you what happens. Let's imagine starting with the top one. We start with the top one, and it turns out that to start a strand of synthesis, we have to make something called a primer. DNA polymerases absolutely require a primer. And what is a primer? A primer is a short RNA. All DNA replication starts with an RNA. A primer that's made by an enzyme called primase. Kind of complicated, right? Primase makes that little piece. And then DNA polymerase, which is the enzyme that does this work, grabs that primer, and then extends it. That's the replication process. That's very easy to see. We start there, and we move it forwards. We're going 5' to 3'. Why don't we do the bottom strand the same way? Because you memorize something instead of learning something. You memorize that all replication occurs 5' to 3', but in order for me to do that bottom strand 5' to 3', could I go from left to right? Can't do it. I've got to start, from right to left, I've got to start from left and move towards the right, right? I can't go from right to left. Because if I did, I would be going 5' to 3' that way, and the strands have got to be anti-parallel. Replication of the bottom one has to go from left to right, not right to left. Whoa. Well, why does it go in pieces? Why doesn't it just start right here and go on out? The synthesis of the bottom strand can only occur when a portion of this gets opened up as a single strand. And what does it take to open it up as a single strand? That's the progression of the top one. As the top one moves further along, more of the bottom one is freed, and primase says, oh, let's start here. And then it starts another one. And then it starts another one. And then it starts another one. So the lagging strand synthesis in pieces occurs because replication can only go 5' to 3', and the strands have got to be anti-parallel. Yes, sir? Lagging strand is always the same size. You mean as the leading strand?
Yeah, well, no, but as each section of lagging.
Okay, so his question is, do these pieces all end up being the same size? Roughly. They all end up being roughly the same size. It's not an exact measure, but they're all roughly the same size. Yes? Okay. This probably is the most complicated part of DNA replication. Okay? If you understand this, you're in great shape. Everything else you can commit to memory and you'll be fine. But if you didn't, if all you did was memorize what I told you about DNA polymerases and you didn't internalize that, that's going to be problematic for you. If you don't understand that, come see me. I've got figures I'll draw out. I've got horrible drawings that I'll make for you and show you how that works, okay? But make sure that you understand why it is that a leading strand is made in a continuous piece and the lagging strand is made in a series of pieces. These pieces have a name. They're named for the person who discovered them, and his name was Okazaki. So we call these guys Okazaki fragments. O-K-A-Z-A-K-I. Okazaki fragments are the pieces that are found on the lagging strand. The leading strand doesn't have a name. It's just called a leading strand. Okay, so that's what's happening in this replication process. So I said something about DNA replication, and I've got about 5 minutes here. I said something about DNA replication requiring a DNA polymerase, but I haven't told you much about that polymerase. So now I want to spend a little bit of time talking about the enzyme that catalyzes all of this. DNA polymerases are interesting and unusual enzymes. First of all, they only work 5' to 3'. There's that thing you memorized. Second of all, they will only continue a strand that has already been started. They will only continue a strand that has already been started, meaning they cannot start the synthesis of a strand by themselves. That's why the primer is necessary. And that's why the primer is RNA, because if it were DNA, that would mean a DNA polymerase was starting a strand by itself. Can't do that. So in this case, we can see here's a strand that has already gotten started, and this DNA polymerase says, oh, look, I can go on to the end of this guy, and I can extend this strand and finish this out and make a double helix. If that top strand were not there, and I added the DNA polymerase and the 4 DNTPs, nothing would happen, because the polymerase could not start that strand. Now, DNA polymerase can extend an RNA strand, It can also extend a DNA strand. And in this case, what I have put on here is a DNA strand. Okay? There are different DNA polymerases that are found in cells. They're not all identical. And their functions are not all identical. Most of the polymerases I will be talking about in this class are from bacteria, because they're the easiest ones to understand. But I will say a little bit about some interesting eukaryotic DNA polymerases. Okay? But what I'm going to tell you about eukaryotic polymerases and prokaryotic polymerases, the basic rule that I just told you, those rules about not being able to start their own, about only working in the 5' to 3' are true for all DNA polymerases. Yes? So a DNA polymerase can extend what's called an RNA primer. If that end of that guy were RNA right there, and it was copying DNA, yes, it would be able to do that. What a DNA polymerase cannot do is it cannot start with an RNA end and copy RNA. It has to be copying DNA. But the end that it starts with can, in fact, be RNA. Yes? Okay? So the DNA polymerase is kind of stupid. It doesn't care how that strand got there that it's extending. It only cares that there is a strand there. There's no strand to extend. The DNA polymerase can't do its thing. Okay? What does DNA polymerase catalyze? It catalyzes the formation of phosphodiester bonds. That's that backbone, phosphate, sugar, phosphate, sugar, phosphate, sugar. The bonds between the phosphate and the sugar are phosphodiester bonds. It's catalyzing their formation. That's what links the strand together. And it's doing it according to the rules of base pairing, which means that the DNA polymerase has to be able to not only catalyze the formation of phosphodiester bonds, it also has to be able to read the other strand and know which base to put in. Now, the last thing I'll say before I turn to the song for the day is a remarkable thing. Okay? The DNA polymerase, even a bad DNA polymerase, meaning one that makes a lot of mistakes, okay, will still be able to do this copying at the rate of about, at a minimum, 100,000 to 1 billion at the high end with only one error. Okay? A bad one makes one error in every 100,000. A good one may go for several billion with one error. You've got 7 billion base pairs in your DNA. Okay? That's pretty remarkable. If we look at bacteria, this will blow your mind. Okay? Bacteria have DNA polymerases that not only are good for about 1 for every 10 million or so, 1 error per every 10 million, they also replicate DNA at 1,000 nucleotides a second. Now I want you to think, if you've ever typed, how many errors you make typing, and just try to conceive of making 1,000 characters in a second, let alone the right ones, right? And that's what DNA polymerase is doing. There's some remarkable things about nucleic acids, and I'll talk about those. But before we do that, I have a song for you about this. It's called Major Groovy. Let's turn to it. DNA forms A and B. A base is complementary, despite similarities differ in the major groovies. Words changed. Na 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 na. Major groovies. Transcription factors with their binding, cause DNA to start unwinding, holding it aggressively by forming bonds in major groovies. Na 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 na, major groovies. For proteins, the key to sequence ID is hydrogen bonding, each base pair unique. Purine, pyrimidine, patterns discrete. And DNA's most major groovy. Na 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 na, major groovy. All right, guys, see you on Friday.